Alright, as a certain prof would say, ladies, gentlemen, other life forms, welcome to today's discussion on Lentic ecosystems by U5. Okay, so looking at the title slide, it probably reflects your brain cells this far into the SEM, doesn't it? Well, never fear, our group is here to guide you through this lesson. You can think of it as a quick crash course outline type of vid to help push you to the more nitty bits that are important as you'll dig through the readings. And I do believe it's tradition around here to start with the last breaker first, get those neurons pumping. So throughout the discussion, you can slow the vid down if we're talking too fast, or if we're asking questions, just pause and try to think of the answer first. Don't worry, there's no wrong answers here, just right ones and intuitive guesses. So let's try this. Between A. Springtails, B. Mayflies, and C. Caddisflies, which group is exclusive to lentic systems? And the answer is A. Springtails. Now, they're a really niche species exclusive to most shallow still waters, and they function as collectors. By the way, this is from Tallinn 1998. It's a really comprehensive read. I suggest you look into it. Okay, let's try something from Baludo et al. So let's say you encounter a lentic system for the first time, like a lake, and you want to get the overall morphology and depth scans. So what would you use? If you thought of an echo sounder, you are correct. So from the name alone, you can tell it uses sound waves to get the outline from the depths of the system. And in case of Baludo, they came with an attached GPS just for good measure. But what if you want lower depth samples for something like a physiochemical analysis, or surface samples for something like a phosphate analysis? Well, if you remember lab, it is indeed a knee skin water sampler. Remember, it's on the wrist and how you let go. But now let's try something a little more conceptual. Algae during the day give off oxygen as part of photosynthesis, supposedly more than what they actually consume at night, right? But they are still associated with eutrophication from increased nitrogen and phosphorus, which still does give us anoxic dead zones. But why? Well, the answer is while light going into the system during the day may actually change or vary depending on other factors, the night consumption of oxygen is relatively the same. And in addition to that, try to remember that bacteria and decomposers use oxygen just to break down organic matter. And also, I want you guys to try to remember something like stratification and depth, and we'll get more into this later. But essentially, too many blooms will block out the sun, preventing it from reaching the lower layers. From there, you get a dead zone with no nutrients, and at worst case, it goes from hypoxic, which is partial, to anoxic, completely devoid of oxygen. So, what a way to start, right? But now I have another question for you that's more personal and less abstract. I want you guys to ask yourselves the question: is it enough to, say, put water into a random area? Let's say you make a simple puddle. Is it enough to call it a lentic ecosystem? Well, it's a bit more complex than that. Remember, the keyword here is ecosystem, right? So you have the basic template established with water, sure. But what about the surroundings? Can external nutrients interfere? Will life burgeon in that water? Maybe how many characteristics can establish dynamics within that system? So these are considerations that lead us to today's topic. And before we leave to the lesson proper itself, I have one more question. What comes to your mind from the word lentic? So lentic ecosystems, guys. From the word lentic, you can already attribute it to still waters from the Latin word lentus, which means sluggish. Already, this would include your marsh, bogs, swamps, ponds, or lakes, and I think it's a good way for us to start with the characteristics. So beginning with physical, I instantly just think clarity, like skin, or movement, like stratification. This stratification, of course, results from differential heating, which forms your differences in density. Better regulation would happen under stronger winds, but otherwise, you have the three usual layers. Why is epilimnion on top? Well, remember that this has to do with water density. More heating means less dense molecules. Your thermocline would form a barrier of intermingling temperatures, while hypolimnion is your cooler and more dead like zones. During spring and summer, though, these layers would basically just form a cadenza of intermixing temperatures and distribution of oxygen throughout the body. While you would also consider suspended solids and light penetration, well, the light would be extended with breach thanks to phytoplanktons. But for suspended solids, these are usually decomposed materials from the allochthonous, as in allocated from outside, or autochthonous, like produced from plants and other algae in the water. So here in the graph, you'll notice that what's interesting is that wetlands actually have better products as opposed to fresh waters. And the reason for that is because you actually have products that first end up as sediments turned in detritus before actually being consumed. And when we talk about size and flow rate, I think these are keys with how a system would cope with pollution. So we say something like resident or retention time, which is your volume divided by outflow. And this tells me that systems can recover naturally, like Lake Erie, but these are under the parameters that A, the source of pollution is removed, and that time is actually allowed for the system to recover with no intervention. Now, with chemicals, obviously from the lab, you'll remember ammonia, nitrate, all that stuff, essentially dissolved gases, right? But given that a lake is indeed eutrophies naturally, this would actually take centuries, but due to human factories and input, it only takes decades. So given that a lake is indeed stratified, would the pH be higher or lower, and in what zone would it be so? Well, if you answered higher for upper layers, you'd be right. This is because hydrogen respiration and decomposition would still lower it. Released carbon dioxide would react with water to form carbonic acid, which could lose one or both of the ions, which in turn releases the hydrogen ions to decrease the overall pH of the water. When we talk biological, on the other hand, I just think of distribution, dynamics, and so on. So from the littoral zone, you would expect more on slope changes and submerged plants, but otherwise you have stuff like crawfish, snails, reeds, in other words, loss of nutrients. While the pelagic or limnetic is more disconnected from the shore or bottom substrate, so it's more like open waters and larger fish. The benthic is actually linked to the substrate, so I would expect more of benthos, mollusks, crustaceans, catfish at this part. So with the two cascade models, very easy bottom-up control. Lower organisms would affect the higher organisms by limiting their supply, and the top-down control would affect them in the way that predators would affect how well the producers would be able to grow and store nutrients for the ecosystem. Which brings us to what I believe is the important part for understanding lentic wildlife dynamics, the PEG model by Summer et al., and this leans more on the ecological side of things. So notice that the figure from the left is actually from a different paper, but I feel like it presents a better visual than the original. So the PEG, or Plant Ecology Group Model, is actually typed for sequential statements describing seasonal events in what would be an ideal lake. Here I have summarized it as follows, so don't worry. Early on, it follows the land seasonal cycle. By the end of winter, light is stronger again, and phytoplankton communities can sprout. This is your first rising slope. Accordingly, zooplanktons would graze and grow as well in population, but the order of appearance for herbivore species depends on how fast they actually grow. Eventually, zooplankton grazing causes phytoplankton population to decrease, while the zoos would rise. Only the inedible ones would remain after to grow, and at this point, the water should be clear. Any other nutrition would be recycled. This plus fish predation would cause the zooplankton population to fall. Now, this in turn, of course, means that the zooplankton's population would rise again with more diversity for the summer. And such a spike, however, would lead to nutrition wars. Phosphate depletion means large diatoms now replace green algae, Silica depletion means cyanophyta now replace your diatoms, and nitrogen depletion means algae now become nitrogen fixing species. Smaller crustaceans replace the larger ones, and now population and variation would fluctuate as it tries to find a balance over the summer. Now, eventually, physical factors become too dominant in the fall turnover, and autogenic succession stops. Eventually, algae will develop again, just not as massive as before. Their return would also harken the decrease of light as winter now reaches another turning point, and the fall of the algal populace. Resting stages will commence until spring would then start all over again. However, do note that the fidelity of the model depends on the actual environment and setting of the lake or Lentic system, which is why, fast forward 2014, Mustaka et al. Have supplied a revised model which works on lower altitudes. So here, winter actually has significant light supplies, so fish predation on zooplanktons is less erratic, which means that your phytos actually have a chance to grow during the season. Here, that it's more like a temperate springtime type of trend, and nutrition restrictions can happen at any point annually rather than the peak temperate seasons. Top down control by fish is also stronger because fish thrive longer in these periods. So, minimum growth would occur in spring, continues by summer, and reaching max biomass by autumn. So, more or less a less erratic trend compared to the original.
Now, let's talk about the different physicochemical parameters and its implication to a lentic ecosystem. These are important to know the tropic nature of a water body. The temperature, much like in lotic ecosystem, significantly affects the rate of chemical and biological reactions in a lentic environment. It affects the dissolved oxygen level in water, photosynthesis of aquatic plants, and metabolic rates of aquatic organisms, and the sensitivity of these organisms to pollution, parasites, and disease. Water temperature is measured through the use of a simple thermometer, a pH probe like the one flashed on screen, and it is usually interpreted as degrees Celsius or Fahrenheit. pH denotes the acidity or alkalinity of a water being tested. Knowing the average pH level is extremely significant to the health of an aquatic life. If a regular pH is not attained, it can lead to the death of aquatic life as some of these organisms are sensitive to changes in pH and a few of them may not be able to tolerate such changes. pH fluctuations can also affect the solubility and toxicity of heavy metals such as aluminum and mercury. Low pH, such as 4 or less, can raise heavy metal quantities. pH is measured through the use of a pH probe, and a pH value of 7 is the neutral. Total dissolved solids (TDS) and total suspended solids (TSS) are two parameters that can increase water temperature and consequently decrease the amount of dissolved oxygen. Both increase turbidity and reduce light penetration that some organisms need to photosynthesize, thereby reducing their ability to produce food and oxygen. Suspended solids can also be detrimental to fishes, as it sometimes blocks their gills, making them unable to properly breathe. TDS is measured by using a TDS meter probe, and gravimetric method can be used to determine TSS. Both are interpreted as milligrams per liter. High chloride content can cause poisoning of our aquatic organisms. It can also result in increased water density that can change the stratification in lakes. Chloride was not usually an alarming pollutant to lentic ecosystems. However, the continuous increase of chloride content globally due to surface runoff and other factors now is near or already exceeds the limit set by governing bodies. Argentometric method can be used to determine chloride content and is also interpreted as milligrams per liter. Higher phosphate content in the lake can also be harmful to marine organisms. It can cause a development of aquatic bloom that reduces the quantity of light in the water and again decreases the amount of dissolved oxygen. Eutrophication causes algae growth. The decay of this bloom will deprive aquatic life of sufficient oxygen. Phosphate can be measured through several phosphate tests. A time-efficient phosphate test kit can be beneficial. It is also interpreted as milligrams per liter. Lastly, dissolved oxygen is necessary for the survival of aquatic organisms. It is the amount of oxygen dissolved from the surface water or as byproducts of aquatic plant photosynthesis. It is one of the most common indicators of life in water, meaning if is too high or too low than the normal, aquatic life may cease to exist. DO is measured through the use of a DO probe and is usually also interpreted as milligrams per liter. There are several probes, equipments that can already be used for measuring multiple parameters at the same time. To apply this knowledge, I would like to share this table from Martinez and Galera in 2011, where they monitor and evaluate the water quality of Taal Lake using the two different physicochemical parameters. The average of two sites were compared with the standard set by DEN or BMB, and from the water temperature to total suspended solids yield that the sample was within the standard. However, the chloride content greatly exceeded the standard, and this can be attributed to the several tributaries that drains their water with high chloride content. Additionally, the phosphate as phosphorus content also exceeded, and Helmer and Hespanhal in 1997 explained that heavy rains brought about the domestic washes containing detergents and topsoils carrying naturally occurring phosphorus from its neighboring highlands. Lastly, the low levels of dissolved oxygen, which can be attributed to severe pollution. This can explain how there are several fish kills happening in Lake Taal.
Now that you have an idea about the general characteristics that make up a lentic ecosystem and the biotic and abiotic factors surrounding it, I will now discuss one of the major roles of lentic ecosystems such as lakes that affects global warming, the methane and primary production in lakes. Methane is mainly produced from wetlands such as lakes and ponds, and another main source of methane is agriculture from livestock, waste management, and rice cultivation. Lakes are important but often overlooked contributors to atmospheric greenhouse gas concentrations, in part due to the emission of methane. Methane is a powerful greenhouse gas that traps heat 34 times more effectively than carbon dioxide. Methane is produced under conditions where little to no oxygen is available. The relationship between lake methane dynamics and trophic status is often attributed to a combination of both extreme anoxic conditions common in productive systems and an increase in autochthonous organic matter settling to sediments fueling anaerobic decomposition. Hence, this methane production in lakes affecting global climate can then influence lakes' community structures. Interactions between climate change and lake biota are complex. Responses are often organism to organism and vary among ecosystems. One major indicator of climate change in lakes is the plankton community. Planktonic organisms have been widely used as indicators of ecosystem change, and because of these relatively short-lived organisms, they respond rapidly to subtle thermal changes. They are also useful indicators of climate change. Phytoplankton and zooplankton communities in many lakes have been relatively well documented over extended time periods, and these records are useful for extracting climate-related responses. Growth rates, abundance, and species composition can each be considered as an indicator of climate change. Climate may also affect species diversity and composition through the invasion of non-native species that expand their geographical range as water temperatures warm. Lastly, the use of lakes as sentinels not only allow us to detect global effects of climate change, but it can also further our understanding of ecosystem processes in lakes. If you want to know more about current studies addressing lentic ecosystems, here are some known and reliable journals that include studies about these ecosystems. But for us to really understand and visualize, we now move to discussing the status and current literatures covering lentic ecosystems here in the Philippines.
To be specific, let us look on the different lakes in the Philippines. Flashed are the 10 major lakes in the Philippines. However, there are a total of 216 recorded lakes as reported in the state of Philippine biodiversity. Because of time constraints, we would like to emphasize on certain lakes, the major and top three largest lakes in the Philippines that have been a target research site by freshwater scientists, which are Laguna de Bai, Lake Lanao, and Lake Taal. Due to their importance, water quality assessment and its flora and fauna are the general centralized topics for research on each lake.
Laguna de Bai is the largest lake present in the Philippines. It provides livelihood to locals present nearby, transportation, recreation, and a source of food such as the fishes present in the lake, and also acts as a domestic water supply. The lake is naturally eutrophic and highly productive. This slide presents the parameters tested in the lake. The presence of nitrogen and phosphorus is highly caused by human activities. There is a variation in pH levels depending on the season, such as the cool months and the midsummer months, wherein it is slightly lower during the cool months. Flash are the average levels of the physical chemical parameters tested in the lake.
Proceeding to the second largest lake in the Philippines, geographically located in central Mindanao. Lake Lanao, a tropical lake and is the oldest lake in the Philippines. Lake Lanao and its catchment basin is characterized with hilly to gently rolling topography. With the deterioration of the lake's water quality, the lake's degradation is now quite obvious, with the data of the lake's existing water condition basing from the chosen parameters flashed on the screen. Anthropogenic activities, on the other hand, threaten to wipe out the species' habitat and population. One example would be the endemic cyprinid species present. Degradation of the lake's water quality has a far-reaching harmful impact on the aquatic ecosystems, survival, and reproduction.
Moving on, the last is the Lake Taal. It is known as the third largest lake present in the Philippines and the second largest lake in Luzon. There is a small volcanic island in the middle of the lake which has been the site of almost all the historic volcanic activity. Ramos 2002 reported that the primary explosion of the lake prompted the initial formation of the lake craters and ignimbrites, resulting in the various caldera present, classifying it as a caldera lake ecosystem. In addition to the previous slide, Lake Taal is located in an agricultural and industrial region that is densely populated and quickly expanding. It is also used for aquaculture, recreational activities, and as a local attraction, demonstrating its economic value. The Sardinella tawilis, giant trevally, and the Lake Taal sisnic, hydrophysim fairy, are almost all the endemic species found in the lake. Overfishing and pollution of the lake by wastewater from businesses and families are two of the most serious issues for this freshwater ecosystem. As for the parameters tested in Lake Taal, we would like to emphasize the difference between the values on the aquaculture and non-aquaculture sites, as aquaculture is rampant on the said lake. It can be observed that in aquaculture sites, the values of phosphates, total dissolved solids, pH, and temperature is slightly higher than in non-aquaculture sites.
Therefore, financial support such as research grants and funding for postgraduate studies and projects are extremely important, along with continuous research efforts to update the status of the lentic ecosystem present in the Philippines. And once again, good day, everyone. We have invited a guest speaker for today's session. So now it's a great pleasure for me to introduce our speaker today, who will talk about her personal experiences and how it's like to be a Filipino limnologist and share some advice for us young undergrads who wish to pursue a career on the same path. A little bit of background with regards to our guest speaker. She is a graduate of the BS Biology in the UST College of Science, took up her master's degree at the same university, and also under the DOST SEI ASTHR Graduate Scholarship during that time. Currently, she is the Senior Science Research Specialist of the DOST JSPS Methane Dynamics Project of the UST Ursinus. She is also just a fascinating person. Ladies and gentlemen, please join me in welcoming Ma'am Justin Tatin De Leon, MSc.
Good morning, everyone. So it's actually evening because I recorded this in the evening. But yeah, good morning. So I'll be talking about basically a how-to of becoming an early career scientist in, of course, the Philippines. Early career scientist is actually not an official title recognized in the country. It's actually a term I just borrowed from first world countries. But I think it was the best term to use to describe what I have basically experienced as a young researcher in the Philippines. So before I get on with my talk, I wanted to share first this meme, which I'm very fond of. So yeah. there are many benefits to being a freshwater biologist. I don't know if you're familiar with this meme, pero and if you're familiar with this group, itong group na ito, but if you're not, I highly suggest joining this Facebook group called Wild Green Memes for Ecological Fiends. I wanted to share this meme because for the untrained eye, parang it's a good picture depicting whatever a freshwater biologist is doing. But for us who has experience in the world of biology, of sampling collection, and stuff like that, may makikita at makikita ka talagang mali. So, in this picture, very obvious na may mali. Si Kuya, si Guy. So, what he's doing is he's trying to observe something dun sa sampling bottle that he is holding. So, the first wrong thing that he got here is that wala siyang makikita dun sa bottle na hawak niya. The bottle is very opaque. So, whatever he's trying to observe will be unobservable. So, kung water samples man yan, bakit mo titignan yung water sample na kinuha mo? If this sampling bottle contains organism like I don't know, zooplankton, you won't be able to observe anything there because of the color of the bottle. The second thing that people from other countries find funny about this picture is that this girl right here is using her laptop. Obviously, not waterproof laptop kasi hindi siya yung types na what we call as rugged laptop, yung waterproof, yung medyo mukha siyang attache case na type of laptop. Yun nga, she's using her laptop beside the stream. So, when I first saw this meme, etong guy lang yung nakita kong nakakatawa. But then when I read the comments, parang they're criticizing this girl. Like they say, bakit di gamit yung laptop niya? Mababasa, masisira. Then I realized in our setting, in the Philippines, in a third world country, this is actually a pretty common thing to do. Personally, I experienced doing sampling while with my laptop. Nasa gitna pa kami mismo ng lake. Well, in the middle of the lake, I'm using my laptop to obtain or to get or collect samples or data sets. So yeah, for other people in the highly developed countries, this is not something that should be done. But in our case, in the Philippines, very common thing to do. And ayun, personal experience ko na rin yun kasi I've been doing research or doing freshwater biology research since 2018, and I've been using my laptop since 2018 in the middle of the lake. So yeah, so a little bit about me. So yeah, before I get started on how to be able to guide you on how to become an early career scientist, so this is just a bit about me. So I'm Justin, better known as Tatin De Leon. So people have been calling me Tatin. If you ask Dr. Papa, your Dean Papa, he, she, he's actually my PI. He'll be referring to me as Tatin. I'm better known as Tatin. If you ask like Dr. Briones, Sir JC, he only knows me as Tatin. So yeah, better known as Tatin De Leon. So right now, my position or my title, my official title is actually a senior science research specialist. So I'm working under a government funded project called I mean government funded project by DOST-JSPS, and we're working on the methane dynamics of a one of the lakes in San Pablo, Laguna, which is Lake Yambo. So I completed both my bachelor's and master's degree in the University of Santo Tomas. So I entered graduate school kasi I found this scholarship that I was very lucky to be accepted at. So, yeah, this is very crucial, which I'll be talking about more later, on why you should consider applying for scholarships when you plan on entering or continuing your studies, your postgraduate studies. So the first question, of course, is how to get started. So for me, this is like a personal parang experience on how I navigated my path and got to where I am right now. So the path I took is that I joined the dark side, basically. So we have this joke na if you're a fan of Star Wars, which I am, yung kumbaga the light side or the Jedis are the ones who are taking up medicine, like med school, and then the dark side is grad school. So yeah, so we have this inside joke sa laboratory na join the dark side. We have cookies. So yeah, join the dark side, give in to grad school. So this is the path that I took. So after completing my bachelor's degree, I entered graduate school to continue with my postgraduate studies to get my master's degree. So basically, this is what I experienced during my 2 years of stay in the graduate school. So my work or my master's thesis actually dealt with copepod ecology. So these are my two species of interest. So this one is an invasive species, so it was introduced in our Philippine waters, and then I compared it to our very own endemic copepod species. So this is me doing zooplankton collection. This is actually just in um, sa North Avenue, yung hospital dun, veterans. This is in Veterans Memorial Golf Center. Yun, so we also do sample collection in like areas in NCR. This is taken during my oral defense. I'm actually part of the first batch who experienced online defense, which is ano ba dito? Parang nalugian ako kasi parang for me, hindi na full yung experience ko being a graduate student. But anyway, I got to defend, I got to pass, I got to complete my degree. So all's good. This is in Lake Buhi in Bicol. This one naman is in, I think, Lake Lumot in Laguna. And then this one is in Sagada, um, Lake Danum, which is now, I think, known as Lake Danao in Sagada area. And then this is Dr. Papa in Cagayan. So, yung dean nyo ngayon, dati bankero ko lang. So, but yeah, he helped me during one of my sampling trips in Cagayan, Cagayan Valley ang tawag dito? The North Luzon. And then this is me, um, na ginulo-gulo ko yung ayos ng mga microscope in our laboratory para matapos ko yung kailangan kong gawin for my thesis. So for my my master's thesis, these are the sampling sites I visited. So yeah, buong Luzon yung vinisit ko. So from northernmost, from Cagayan until up to Bicol region. I also did one in Mindoro. Kasi may major lake doon si Lake Nauhan. Nauhan daw ang tawag sa kanya. So, like I said earlier kanina, I wanted to highlight the importance of finding scholarship when applying for postgraduate studies. Kasi ito yung importante na part dun. If the type of study you're going to do, or your master's or PhD thesis you're going to do is not going to be part of a project, you need to be self-sustaining. So, ikaw yung gagastos ng lahat ng kailangan mo. So, personally, I advise those people who want to go study graduate or want to enter graduate school by looking into scholarships they can apply to. For me, I applied for a DOST graduate scholarship. So with their help, meron nabayaran ng buo yung tuition fee ko. I get paid monthly to complete my study, to complete my degree. And then they also gave me funding to do my thesis. So yeah, but I also applied for other funding for other grants outside the DOST provided na what do you call this funding. So I also applied for an international body para mabigyan ng further support yung study na kinokonduct ko kasi ayun nga, isa buong Luzon yung study na kailangan kong gawin and it didn't end there. Sampling, may mga analysis part pa syempre na kailangan kong gawin. And traveling is not the only thing na nagko-cost naman, pati syempre mga materials mo, yung lodgings, and all that. So with that, I wanted to share ano ba ang mga experiences ko or observations doing research in the Philippines. This is like a combination of what I experienced during my graduate studies and as well as what I experience now as a researcher of our laboratory. So these are the things that you need or you can expect that will happen. So when you're doing your research in the Philippines. So permit application involves a lot of bureaucracy. I know from what I remember, I think you have experience parang discussing permit application during one of your courses. I think I heard Sir Peralta discussing it like that. Pero ayun, there's going to be a lot of bureaucracy when you apply for a permit kasi part yan ng fieldwork process nyo, pre-fieldwork, you need to apply for permits. So may mga places na you can just simply contact the city hall, parang tatawagin mo lang sila, pupunta ako ng ganitong oras, ng ganitong araw, sa mga ganitong lugar. But there are also places that you need to show up physically to apply for a permit. So I conducted this type of visiting when we conducted fieldwork in Bicol. So I had to visit like 4 municipalities in one day. Kasi syempre tagtipid ako noon. So I had to finish it in one day and then go home. So I visited like 4 municipalities in Bicol to apply for permits in the different municipality hall. Ayun, para lang mag-conduct ng sampling sa Buhi, sa Baaw, sa Bato, and then I forgot the other lake, but we visited 4 sites. Danao. Lake Danao in Bicol. So, yun. Next is fieldwork in freshwater biology is manual labor. Hindi ko sinasabi na kailangan mo maging fit talaga to conduct field sampling. Pero you have to expect that mapapagod ka talaga. You'll be the ones carrying the equipment, carrying your samples to and from yung sasakyan mo. May mga times na you have to trek mountains or hills bago ka makarating sa sampling site mo. And then you have to go down the mountain with like 3 liters of water in your back. Diba? Yung mga kinolect mong samples. So, yeah, fieldwork is manual labor talaga and expect mong mapapagod ka talaga when you're doing fieldwork in the Philippines.
Flashed are the references used.
With that, we hope that you all learned something from us today and enjoyed listening to our special guest speaker. We hope that Ma'am Katin's personal experiences as a limnologist inspired you and that this information will be helpful to us, particularly to those who aspire to continue in the graduate school. If you have any additional questions for Ma'am Katin, please feel free to message us and we will send your questions or concerns to her personally. Thank you again and have a great day, everyone.